myself Lokesh. I am from group 317. My topic is on the properties and physics of the proteins. So, relationship between the primary and the three-dimensional structure of the proteins. The relationship between the primary and the three-dimensional structure of protein can be illustrated by the four main stages of protein organization. The first structure is primary structure of protein, which is also called the amino acid sequence. This is the linear sequence of amino acid in the polypeptide chain. Each amino acid is connected by peptide bonds. A specific order of this amino acid is determine the proteins and their properties and how they will be folded. Examples of proteins are hemoglobin, valine, histidine, leucine, threonine, proline, and glutamate. Now the secondary structure. It is also called the local folding patterns. Polypeptide chain begins to fold into the repeating patterns like alpha helix and beta sheets. In this structure are stabilized by the hydrogen bonds between the backbones atoms of amino acids. The examples of secondary structure is alpha helix and beta plated sheets. Now the tertiary structure, it is also called the overall 3D folding. In this, the full three-dimensional structure form has interactions between the side chains. Functional groups of amino acids occur. In this structure is stabilized by various forms including the hydrophobic interactions, hydrogen bonds, ionic bonds, and disulfide bridges. Examples of tertiary is alpha keratin and carboxypeptidase amino acids. The fourth one is quaternary structure. It is also called the multiple subunit assembly. In proteins made up of more than one polypeptide chain, the quaternary structure describes how these chains are arranged in relations to one another. Examples of quaternary structure is hemoglobin, DNA polymerase, ribosomes. The three-dimensional structure of the protein often referred to as its quaternary structure has the overall shape that as protein that tubes over folding its linear chains like primary structures. This shape is crucial because the functionality of the proteins is linked to its 3D structures. Here the breakdown of these aspects involved in the formation and importance of the three-dimensional structures. The tertiary structure formation. It forms as a result of interaction between the amino acid side chains and R groups in the proteins. This bonding and interaction take place by several steps. The first one is hydrogen bond, formed between two polar side chains. The second is ionic bonds, occur between opposite charge side chain. The third is hydrophobic interaction, non-polar side chains and together inside the protein. Van der Waals forces. These are the weak attraction between the and closely packed atoms. Disulfide bond. It is the strong covalent bond between the sulfur atoms and crystalline residue. Second is domains and motifs. Larger proteins often have domains which are distinct functional and structural units within the proteins. Each domain can fold independently. Motifs are small structural elements within the tertiary structure such as helix loop helix motifs or zinc finger, which are involved in particularly biological functions like binding DNA or other proteins. Third is the stability of the 3D structure. 3D structure of proteins is generally stable but can be disrupted by the factors like temperature, pH changes, and chemicals, a process known as denaturation. Denaturation can cause the proteins to lose its functionality. Fourth is functional importance. Active sites. In enzyme, the 3D structure creates pockets, bears as active sites, proteins interactions. The 3D structure allows proteins to interact specifically with other proteins or molecules. The binding specificity. The shape of protein allows them to recognize and bind to the specific molecules with high specificity such as hormones binding. Fifth is folding pathway. The folding of proteins into their correct 3D shape is usually a highly regulated process. Like chaperones, proteins often assist other proteins in achieving this correct folding, preventing misfoldings. The sixth is quaternary structure. In some cases, multiple polypeptide chains came together to form a quaternary structures like hemoglobin, which consists of 4 subunits. The conclusion of all that is the end of the three-dimensional structure of a protein as vital to its biological functions. These structures result from a folding of the polypeptide chain driven by interactions between the side chains of amino acids. The proper 3D structure is critical for binding, catalytic activities, and overall protein stability. My second topic is the structure and stability of the globules. The study of electrophorosis which give information on charges on the globule. Example, the study of denaturation and proteolysis which field data in the stability of the globule. The first one is myoglobule and the hemoglobin. The structure of this protein has been studied in detail by X-ray diffraction methods. The myoglobin molecules which contain the prosthetic heme group is built up by 8 alpha helical proteins intersected by disordered portions. In contrast of myoglobin, the hemoglobin molecule has a quaternary structure. It is composed of 4 chains coiled into globules, 2 chains designated as alpha and 2 is chain is beta. The structure of each of the alpha and beta globules is similar, so the structure of myoglobule. A detailed analysis of the structure and myoglobule and hemoglobin showed that the cores of their globules are really filled with tightly packed non-polar side groups, aldehyde of the amino acid residues. The hydrophobic interaction. Hydrophobic interaction, it play a most important role in the self-assembly of the globules. The secondary structure of the alpha helix portions are also determined by the hydrophobic interactions. The electrostatic salt bridge in proteins are also stabilized by the water environment, since their formation leads to the release of the orientated water molecules. Denaturations. Denaturation means the destruction of these periodic crystals. As a result of the destruction, we obtained averaged information on the stability of the internal bonds and on the electronic conformational interactions. The denaturation of a protein by an acid or a base is directly associated with the electrostatic interactions. The free energy of a polyelectrolyte coiled into globule is proportional to the square of its surface charge. The titration with an acid or a base can ionize the internal groups, which enhance the attraction of the hydrate cells and shifts the equilibrium to the coil. Thank you.